Now we'll take a look at transport across membranes. First, we'll look at passive transport. Remember, passive transport means that the cell does not spend energy to move materials across this membrane. The simplest type of passive transport is diffusion. In diffusion, molecules will move from an area of high concentration to an area of lower concentration. That is called moving down the concentration gradient. This figure shows that where we are going from a high concentration to a low concentration, going down this gradient. Substances want to move in that direction, so it doesn't really require energy to do that unless there's some barrier preventing them from moving. In membranes, this movement will typically occur through this lipid bilayer. So these molecules, if they can fit through the membrane bilayer, will travel across there. If they go through the membrane bilayer, we typically refer to it as simple diffusion. So often when we use the term diffusion, we don't even need a membrane. If you put dye into a beaker of water, those molecules will diffuse throughout that beaker. When we are looking specifically at transport across membranes, we'll sometimes add the word simple in front of that to clarify that this is diffusion across a membrane and it's typically going through this bilayer. And those molecules will move until they reach equilibrium. So here we have a high concentration in the extracellular fluid, very low or little concentration inside the cell. These molecules start to move, and they continue to move until they reach this equilibrium here on the far right. Now, at equilibrium, they're still going back and forth, but essentially for every molecule that comes in, another one goes out, and there's no net change across that membrane. The only types of molecules that can really move this way are typically small nonpolar molecules. Oxygen, carbon dioxide can move that way. And then some of the lipid hormones, because they can actually dissolve into this lipid bilayer, can move this way. Most other molecules can't. So over here we see this passive transport, these molecules like oxygen and CO2 going through that bilayer from an area of high to an area of low concentration. So this is our simple diffusion. There's a variety of different factors that affect diffusion rates. One is the extent of the concentration gradient. The greater the gradient, the faster the diffusion. In this image, we see that the gradient, or the difference in the concentration between this box and the top one, is not very great. So that's a low gradient. We don't have a big concentration gradient there, not a big difference. If we look at the bottom one, we have a big gradient. So there's a big difference in the concentration between those two. If there's a bigger gradient, then the diffusion rate is faster. So that's why we have this arrow bigger, because we have more diffusion this way because of the difference in the gradient. Another thing that affects diffusion is the mass of the molecules. If you imagine that what's happening in diffusion is molecules are constantly in motion, they're bumping into each other and spreading out. And so the more the molecule moves, the greater the rate of diffusion, because they will move away from each other faster. And if you think about it, small molecules will move faster than large molecules. That means that small molecules will diffuse more quickly. So in this image, we see large and small molecules here, and we see that these small molecules are moving faster over to this side than are the large molecules. So the smaller molecules diffuse faster. Temperature is another factor that will affect diffusion rate for the same reason we mentioned before. If you heat something up, then the molecules in that substance move faster. If they move faster, they spread apart faster, so they will diffuse more quickly. If the temperature is higher, the molecules move faster, they diffuse at a faster rate. On the top one, we are showing something that's colder, and therefore the molecules are not moving over to this other area as fast. The bottom image is showing a warmer substance, and it is moving more quickly with a faster diffusion rate. Other factors that affect diffusion: surface area. Remember, surface area looks at the amount of area that we have here on the surface of, say, the plasma membrane. So this image is trying to show a cell with less surface area. Here, where we have the jagged edge, has a higher surface area. The more surface area you have, the more molecules can move across that membrane. There's more openings in the membrane. There's more area for it to move across. If we have lower surface area, we'll have a slower diffusion rate. If we have higher surface area, we'll have a faster diffusion rate. The solvent density will also play a role. Dehydration would cause the cytoplasm density to increase. So imagine again, molecules are moving around, and if they're moving in a more dense substance, they're not going to move as rapidly. And so that's going to slow down the rate of diffusion. Distance traveled will also affect this. If we have a system where the molecules have to move farther, then we're going to have a slower rate of diffusion into this farther area than we would to an object that's closer. This is another thing that limits cell size. Remember, we talked before about how cell size is limited by the surface area to volume ratio. Well, one of the problems with a large volume is that molecules have to travel further distances and that the diffusion rate is limited by that. Solubility is another thing that affects diffusion rates. More nonpolar substances, things that are soluble in lipids, tend to diffuse at a faster rate than the more polar substances or charged particles. Pressure is another factor that affects diffusion rates. Imagine having a balloon that's your cell, and it's got pores in it, and you've got substances in there, and now you're going to squeeze the balloon. And if you squeeze the balloon and put a lot of pressure behind it, more will go through that membrane than if we don't put pressure behind it. This happens in our kidneys. When the blood goes into the kidney, it's under pressure, and it causes a lot of molecules to move out and to diffuse out. This graphic is showing a cell that is not under pressure. This concave membrane here lacks pressure. This convex membrane here has a lot of pressure to it. So where there's not a lot of pressure, we get very low diffusion, and when there's a lot of pressure, we get a fast diffusion rate. Now let's take a look at facilitated diffusion. In facilitated diffusion, it's still a passive process, so it doesn't use energy. The difference here is instead of it going through that lipid bilayer, it will use membrane proteins. It's still going to go down the concentration gradient. If it's a passive process, it has to go down the concentration gradient. You can't have a passive process and move something up a gradient from an area of low to higher concentration. It just won't do that. That requires energy. It's like pushing something up a hill. You have to spend energy to do that. It has to go from the high to low concentration. The difference here again is that it's going to use membrane proteins. Many ions and small polar molecules will move this way. Even though they're small, because they're charged if they're ions, or partially charged because they're polar, they won't go through that lipid bilayer. Remember, the inside of that lipid bilayer is nonpolar, and so those polar molecules get essentially repelled from that. So in order to go through there, we have to have a membrane protein that has some polar sections to it that allow this polar molecule or these charged molecules to go through. We have two different types of proteins that will facilitate transport: channel proteins and carrier proteins. Down here we see a picture of a channel protein, which is essentially just a protein-lined pore in the membrane. And then here we're seeing some carrier proteins that will actually shuttle these molecules across the membrane. Let's look at the channel proteins. The channel here that allows for the passage of the material is typically going to be polar sections of the molecule, and the top and bottom will also be polar. And what that will do is attract ions and polar molecules to it. Some of these channels are open all the time. Others are what we call gated channels. They open and close. When a signal is received, they can open and then they will close. Some important examples of channel proteins: aquaporins are specific channels through which water passes in and out of the membrane. These are very specific to water. Muscle cells have gated ion channels. When a signal is received on the muscle cell, opens the channel and these ions will rush in, and that initiates muscle contraction. Let's take a look at carrier proteins. Carrier proteins will typically be very specific to a single substance. One of the reasons why they're so specific is they bind to that substance. So you have a protein that has a particular binding site. This molecule comes in, binds to it. It causes this carrier protein to change shape, and that ends up allowing it to move that material into the cell. Here we have this molecule comes in, it binds to a location on the protein. When it does bind, it causes a change in shape of that protein, and that change in shape then brings this molecule to the inside. Imagine that it's got arms on both sides of the membrane, and when this molecule comes and binds to it, it kind of flips over in the membrane, and the hand that was holding it is now on the other side. So it just flips it across the membrane. Now, some of these allow movement either in or out of the cell, and then others might be unidirectional, only allowing movement in one direction. But a lot of times they will allow movement in either way, just depending on the concentration gradients. Once again, this carrier protein is not moving things against its concentration gradient. At least right here it's not, because this is facilitated diffusion. We're not spending energy. So this change in this membrane protein was not something that required energy to change the shape. It changed shape, and now the material's on the inside rather than the outside of the cell, but energy was not required. An important example of a carrier protein that does this facilitated diffusion is the glucose transport proteins that bring glucose across the membrane. And we'll take a look at this video that shows us this process.
When glucose is ingested, it is absorbed into the bloodstream. Here, it will be taken to cells where it is used to fuel cellular processes. In order for glucose to be used by cells, it must first pass through the cell's plasma membrane. Transportation across the membrane is facilitated by a family of proteins called glucose transporters, or GLUTs. GLUT1 was the first member of this family to be described. GLUT1 is a membrane protein composed of 12 transmembrane helices and 4 intracellular helices that act as a gate. Upon glucose binding, transmembrane helices 1 and 7 rotate inward. The intracellular gate opens. Glucose is released into the cell, moving naturally down its concentration gradient.
Now let's take a look at osmosis. Osmosis is the diffusion of water across a membrane. We've already talked about how there are these aquaporins, which are these protein channels in the membrane. Why do we have a special term for just the movement of water across a membrane? Well, we know that water is critical to life, and we know that essentially cells are fluid-filled sacs that are bathed in fluid. So for the most part, this diffusion of water is hugely important to cells. This movement of water is critical to the way that cells work. Water will always move from an area of high water concentration to lower water concentration. Now you have to be careful with this definition, because normally when you define concentration, you're thinking of something being more concentrated if it has more solute in it. So if it has more salt, you would think it's more concentrated. And it is. It's more concentrated with salt. However, if it's more concentrated with salt, it's also less concentrated with water. So this is going to go from something that has a lot of water, a higher percentage of water, to a lower percentage of water. This diagram down here shows it nicely. So over here on the left, we have a high water concentration, low salt concentration. On the right-hand side here, we have a high salt concentration and a lower water concentration. Once again, think of these as percentages. In cell membranes, because of their selective permeability, usually water can pass through the membrane because of these aquaporins, and some water can fit between the lipid bilayer molecules. But the other molecules across the membrane typically can't move through the membrane. The membrane is much more permeable to water than it is to the other things that are on either side of the membrane. What that means then is water can move, but the solutes, the things that are dissolved in water, can't. And that's what they're trying to show here on this right-hand side with the beaker. What has happened is water has moved from an area of higher water concentration over here on the left to the right. So now we have more water on the right. And if we were to look at the overall concentration of solute to water, the percentages, now they're the same on the two sides. But what has happened is we've got a net flow of water coming over to this side. And then what that ends up doing is it causes a change in the pressure on the two sides here. So if this were a membrane, this side right here might have pressure built up in it compared to the side over here. So we look at the movement of water because water tends to create that equilibrium in a situation where the molecules, the solutes that are dissolved, cannot do that. Only the water can actually do that. Let's take a look at this video on osmosis.
Before we take a look at how osmosis and tonicity affect a cell, let's review what each of these terms means. Osmosis represents the diffusion of water across a semipermeable membrane. The term tonicity refers to the relative solute concentration of two environments separated by a semipermeable membrane. In other words, by comparing the tonicity of the solutions, you can determine the direction in which osmosis will occur. To demonstrate how tonicity affects a cell, let's place some red blood cells into a beaker containing pure water. In this case, the solute concentration is greater inside the cells than in the surrounding water. In other words, the contents of the cells are hypertonic in relation to the hypotonic contents of the beaker. Because of this, osmotic pressure results in the diffusion of water across the membrane and into the cells. Over time, if enough water enters the cells, the cell membranes may burst. This is called lysis. Now let's place the red blood cells in a beaker containing a solution of salt, such as sodium chloride. Since the contents of the beaker are hypertonic in relation to the interior of the cells, the water within the cells will diffuse across the membrane and into the contents of the beaker. This causes crenation, or shrinking of the cells. If the red blood cells are placed in a beaker whose contents match the tonicity within the cells, then there is no net gain or loss of water. The environments within the beaker and inside the cells are said to be isotonic or the same. An important thing to remember is that osmotic pressure always causes water to move from a hypotonic environment toward a hypertonic environment. In other words, water moves toward areas of high salt or sugar concentrations. This simple process is used to drive the operation of our kidneys and explains some of the physiological consequences of diseases such as diabetes.
Now let's take a look at this term tonicity that was mentioned in the video. Tonicity describes how fluid will move across a membrane when you're looking at a cell surrounded by a membrane that's bathed in a fluid. How will that cell behave and how will that affect the volume of the cell? It's typically correlated to the osmolarity of a solution, which is really looking at the total dissolved solutes. We might have salts and we might have proteins, we might have sugars, and if we total all of those up, we have some measure of the osmolarity. And if two solutions have different osmolarities, different concentrations of solutes, then water is typically going to move. The proteins are too large to move through the membrane. Most of the ions and other molecules that are there won't cross the membrane, but the water will. And so water is going to be the one that moves. So we use these terms hypertonic, isotonic, and hypotonic to describe the comparison of two different solutions or to describe a cell and its extracellular fluid. So hyper, remember, means more. So there's going to be a higher concentration. Iso means the same, and then hypo means below or less than. If we look at a hypertonic solution, imagine you have a cell bathed in fluid that is hypertonic to it. So it has a higher solute concentration than that cell has. The cell is considered hypotonic to the solution, and the solution is hypertonic to the cell. We have higher solute concentration out here, it's hypertonic. We have lower solute concentration inside the cell is hypotonic. Water wants to go where there's a higher water concentration to a lower water concentration. So right now there's a higher water concentration inside the cell, and water wants to leave the cell and go into that more highly concentrated solute solution surrounding it. So water will leave the cell, and these cells will typically shrink up and undergo crenation. If we have an isotonic solution and an isotonic cell, it means they have the same concentration. So the water solute concentration is the same outside the cell as we have inside the cell. And so we're not going to have any net movement of water. Yes, water will come in, but water will also leave at the same rate. And so we'll have an equilibrium there, and there won't be any net change. In a hypotonic solution, we have a lower solute concentration, so the outside of the cell is really now more watery, and the inside of the cell has more solute in it. It has a higher concentration, and it's less watery. Water wants to go from where it has a high water concentration out here to where it has a lower water concentration inside the cell. So the outside of the cell is described as hypotonic, the inside of the cell is described as hypertonic. And water will flow into the cell. When water flows into the cell, it can often cause that cell to first swell up, and then if the membrane can't hold that extra pressure, it can burst. And we call this lysis. And if it's lysis in a red blood cell, we sometimes called it hemolysis. Lysis is to break open, and hemo refers to the blood cell. Animal cells function best when they are in an isotonic solution. So when we talk about regulation of water, we often refer to this as osmoregulation. Let's take a look at osmoregulation in a plant cell as opposed to the red blood cells we saw. The difference with a plant cell is that it has a cell wall around it. And because it has a cell wall, the cells themselves are not going to burst and they're not going to shrivel up the same way that our red blood cells did. Organisms that have cell walls, like plants, fungi, bacteria, some protists, they typically will prefer a slightly hypotonic solution, so a somewhat less salty solution than the inside of the cell. This shows what happens in a typical plant cell, or any cell really with a cell wall, when we place them in these different solutions. What ends up happening in these cells is it changes the pressure of the plasma membrane against the cell wall. So if I start off looking at a cell that's isotonic over here, we have that large central vacuole that we saw inside of plant cells, and it's filled with a lot of water. And normally it's pushing slightly against this so that the cell membrane is up against the cell wall. The pressure of the cell membrane against the wall is also called turgor pressure. And that turgor pressure is critical in organisms like this for growth and proper function. If I put a plant into a hypertonic solution, so I bathe these cells in salt water, the water will leave the central vacuole and leave the cell, and it will allow this membrane to pull away from the cell wall. It's called plasmolysis, where the plasma membrane has pulled away from that cell wall. Over here, where it's hypotonic, we see that the vacuole becomes a little bit too full of water because excess water is coming in, and it pushes that cell membrane tightly up against the cell wall and puts some pressure on that cell wall. So in plants, if they don't have enough water, we lose this turgor pressure. It causes visible wilting. If we give them water, it will increase that turgor pressure, which is what we see here on the right. A protist that's in fresh water is typically going to have a higher solute concentration inside the cell. It's got sugars and proteins and ions and lots of other things inside that cell. This type of a cell then is constantly being bombarded with water coming in. What freshwater protists have to do is use these structures called contractile vacuoles that will fill up with water and then sort of explode at the surface of the cell, pushing that water out. So it's sort of bailing the water out of the cell. Marine invertebrates typically have internal salt concentrations that more or less match their environment. They don't have as much of an issue of maintaining this water balance. With fish, it really depends if they're freshwater or saltwater fish. So you can imagine a fish that is in freshwater is constantly taking in water, similar to the paramecium here. They have kidneys that produce urine that is very diluted. They produce a very watery urine, which ends up ridding the body of this excess water. If they're in a marine environment, they have the opposite problem. They have to get rid of salt and conserve water because they're constantly losing water to their environment. And so now they have kidneys that produce a very concentrated urine that gets rid of salt and conserves water. And there's osmoreceptors in brain cells that monitor the salt concentration in the blood that then affects the function of the kidney. If these osmoreceptors tell us that the concentration of the blood is too high, there's too much salt or too much sugar, then what that would do is to tell the kidneys to conserve water. If the osmoreceptors indicate that there is a low solute concentration, then we might produce a more dilute urine to get rid of the excess water. So that takes us through passive transport. In our next video, we will look at active transport.